I'll just say a couple of words, if you don't mind, before we start. So I really wanted to help our institution with a little bit of focus on quality and safety, and we do a huge amount of lift in this arena in our department. And I thought it'd be a great chance to have Michelle Dahl, one of our star ID physicians, present on a very important hand washing initiative. So I'm going to ask her to lead off, and we are very grateful to our infectious disease division and to you, Dr. Dahl, for all that you do for us and our patients.
Thank you very much. I'm just going to pull up 3 very brief slides, and I will be very brief, not to take away from Dr. Celi's talk today. But as you know, the hospital has embarked on a hand hygiene reminder technology system. And the question we get along is, why did the hospital feel that we need to make this move? Why have we done this? And the reason is that we, after years of looking at our observer data, we're just not happy to be at 80% compliance across the organization. It is just not, quite good enough. And so this is an effort to improve hand hygiene compliance, but also to improve transparency around our practice of hand hygiene, not only for each other, but also for our patients and their families. We know from an extensive research body that patients desperately want their providers to wash their hands, but they are not going to ask us to do so in general because it's too uncomfortable a discussion to have. So from my perspective, straddling infectious disease and infection control, there really is no other intervention that is as closely tied to preventing transmission of organisms, both the ones we do surveillance for, multi-drug resistant organisms, and everything in between, than is a consistent hand hygiene practice. So how have we been doing with this so far? So we were doing quite well. We are implementing, as you know, this project in phases. So we were in phase 3 when COVID hit. And with COVID and the stresses in the system related to that, we did see users of the badge fall. So that graph is the bottom one. On the right side, you can see that through the COVID, when COVID hit the hospital, our users fell. And that was for a variety of reasons. We had issues with supply of the product, as you know. There was a misconception that you could not wear the badges anymore to be compliant with your COVID-related PPE. So just to clarify for this group, you can absolutely wear your badge with your regular badge in and out of whatever room you need to go into. It is going to be under the PPE with your other items. And we also saw that with the pause essentially in this program due to COVID, a lot of providers have misplaced their badges. So if you at this point are realizing that you have no idea where your badge is, please reach out to Joe. The vendor has given us some extra badges to allow us to rebadge individuals into the system. Not a reason that you need to go without. We can just give you another one. Please reach out and we can do that. And then lastly, I wanted to give you a sense of this is the kind of data that the hospital leadership is looking for. As you are probably aware, this is part of our official hand hygiene policy, is participation with the hand hygiene technology system. And this is the kind of data that we will be sharing moving forward. So you can see, in addition to compliance, and we're asking leadership not to obsess about the compliance. At this point, what we're really looking at is the number of users per month from each department. Indicating, participation with the system. So the number, we know that in each division, not everyone is on service at all times. There's a minority of providers who are badged who are going to be having data for that particular month. So we had reached out to each particular service to get an estimate of how many data points do you expect to have for your group. So if you are a leader of these and are looking at this numbers of expected users and see anything way off, again, now is the time to let us know. But this is what was reported back by department administrators and the leaders that we have reached out to as a best estimate of a user rate for each group by the month. If you have, I'm going to stop there and let you get onto the academics. If you have any questions, though, related to what we shared, please reach out either to myself or Joe Armstrong Novak, who is the project coordinator for this initiative.
Super. Thanks very much, Dr. Dahl. And what I recommend is we will send out these slides to the division chairs and they can forward it to their faculty, so everyone can see this and we can make sure we've got the right expected number of users and so on. So I appreciate that, and you know, it's very important data, and thanks for coming at short notice.
Thank you so much.
Great. So now we will move into the more formal part of the program. So it's really a great pleasure to have our colleagues, including Dr. Celi. As you know, Dr. Celi is the chair of the Division of Endocrinology, Diabetes and Metabolism, and we're very lucky to have him here. He's both a fantastic clinician, wonderful educator, administrator, and an NIH-funded researcher with, you know, really huge expertise in thyroid hormones and metabolism. And there's going to be a duet done today with another very exciting young researchers, Dr. Bin Nai, who's a PhD instructor in our department. And they are going to talk today about translational studies on adaptive thermogenesis and its role in energy and carbohydrate metabolism. And I think Dr. Celi is going to also introduce Dr. Nee a little bit further. So thanks, guys. We're really looking forward to this update.
Good morning, everybody. Good afternoon, rather. And you probably know me as somebody who tends to be very vocal and preaching the gospel of diabetes management and leave the clinicians alone in clinic. Well, this time I put the mad scientist hat, and I'm delighted to present what we do in a translational environment. Can I have some thumbs up if you see the screen? And also, again, it is my honor sharing this presentation, and actually having the bulk of the presentation done by Dr. Nee, with whom I've been working since the NIH time, and over the last 6 years, we've been working in developing a translational program here at VCU. So, in other words, the question is, what does a thyroid dude like me has to do with brown adipose tissue? And hopefully, in the next few slides, I can explain what we're doing, why we're doing that. So those are the educational objectives of today's talk. And again, you'll hear over and over the term thermogenic fat. Thermogenic fat, which is a relatively understudied topic, but hopefully by the end of the talk, you'll be excited as well. So first of all, we would like to lay the ground on our research questions. So what is translational research, what it really means, then review the energy balance components in human and rodents, and that's very quickly why we are looking at this question. And then getting into thermogenic fat, whether it's relevant or not. I will present some previous studies from our group on clinical observations leading to detailed studies on thermogenic fat. And then I'll be more than happy to have been taking the baton and going on experimental tools and sharing some what we find exciting findings on role of androgenic factors on thermogenic fat function and expansion. And then I close back to bedside. So what, in essence, what the grant reviewers always ask, the so what question. So translational research, what does it mean? It's really in the idea of the beholder, but it definitely looks cool. And you say, oh, you know, I'm a translational researcher. Oh boy, what does that mean? Well, to me, it's exploring the relevance of basic science and mechanistic observation in humans and in clinical settings. So in essence, is it important or not? And then determining the molecular mechanism underlying clinical observation. So why do we see what happens at the bedside? So in essence, asking the why. And then moving next, it's exploring the potential therapeutic use of basic mechanistic observation. So can we make a drug? Can we make an impact in the disease state? In other words, it's what an MD does. It's an MD that does research. So we're very aware of one ongoing epidemic, but what happened on the other epidemics? So the obesity epidemics, what's the story? And here I start with, as you know, I don't like states going red, so I use a different metaphor. And in this case, I like to show a cat, which is lean and mean, circa 3000 BC. This is an Egyptian cat, and as you can see, it's definitely slender. And this cat has the same genetic composition of this orangey cat who's hopefully going to be evicted at 12:01 on January 20th, and really indulges in fat food, in fast food, okay, call it. And again, this is to show that obesity is a clear example nature and nurture, because the genes are the same, but what changes is the environmental factors. And everybody is looking at a different take of why things happens. And the analogy of the blind people trying to define an elephant comes very apt here. So each in his own opinion, exceeding stiff and strong, though each part was partly in the right and all were in the wrong. So in essence, we really need to look at things in a more comprehensive factor. And we can dissect and reverse engineering what happens in obesity. Well, everybody has their own kind of preferred pathway, and in any grant, the first line is the XYZ pathway is crucial for. Good. And in our case, what we want to say is that it's probably crucial for, and we are interested in thermogenic fat. So adaptive thermogenesis, what is adaptive thermogenesis? In a word, adaptive thermogenesis is the excess of energy that is burnt to maintain the core temperature when individuals are exposed to a temperature which is below the so-called thermoneutrality. For humans, thermoneutrality is everywhere within 28 and 30 centigrades, which is roughly 80 Fahrenheit or higher. And to us is a potential target for energy metabolism modulation. This is a relatively young topic of interest. Everything exploded in 2009 with 3 New England Journal of Medicine papers that showed that individuals who had brown fat, and this is an individual who was exposed to pretty much sitting on a block of ice, and all of a sudden you can see on PET that there are multiple areas of uptake. And when you correlate with BMI, you see that brown fat activity is higher in the leanest individuals. There aren't that many people with a BMI less than 25. I'm sorry to inform you, as well with a low body mass index, a low fat percentage. But that's what's going on. And the question is, so what? Is it relevant for real or not? And as it happens, I took an interest in brown adipose tissue from a very thyroid-centric standpoint. Because from my personal perspective, brown adipose tissue has the characteristic of creating UCP1. UCP1 is uncoupling protein 1, which is a protein that migrates from the cytoplasm into the mitochondria and literally uncouples the oxidation and phosphorylation, dissipating energy into heat. How does it work? Well, the driver is the adrenergic neuron that stimulates beta-3 adrenergic receptor, which then increases the cyclic AMP within the cells. driving the transcription of type 2 deiodinase, which happens to be the enzyme I spent a good 10 years of my life on. And this type 2 deiodinase converts T4 into T3 in a tissue-specific intracellular manner. And T3 happens to be the main driver of the transcription of UCP1. So from my personal perspective, brown adipose tissue is the perfect target to study the effects of local conversion of T4 into T3. And that's how I got into the business. So, but the question is, does it play a metabolic role? So if you're a patient with head and neck cancer or thyroid cancer, having this uptake can be very disconcerting. And this is a clinical observation with BACA-DNAH, in which a woman with type A insulin resistance, so in essence, this woman has no insulin signaling, developed a very aggressive form of papillary thyroid carcinoma. And as it happens, it was a very interesting observation because in the interval of 30 months after the diagnosis, all of a sudden her insulin resistance became incredibly better. And a picture is worth a million words. Please note the amount of insulin the patient was requiring as patients with extreme insulin resistance, 3,000 units per day, yes, 3,000, with a poor control, with an A1C everywhere between 9 and 10. In 30 months, the patient went to 0, just on metformin, and her A1C was smack normal. So now the question is, okay, this patient, is it true correlation or is it true causal or just pure association? As it happens, we treated the patient with radioactive iodine on withdrawal. So this is where she was on high-dose levothyroxine. We repeated the PET scan and the patient had 0 uptake while she had no thyroid hormone on board. And then when we started the high-dose T3, so to kick back after the treatment, all of a sudden her uptake went up again and she became euglycemic again. We then performed biopsies and this is the suprascapular fat, and that's what we looked as a control negative, so subcutaneous adipose fat. And they all look the same and they all look like a brown adipose tissue. How do we know it's brown adipose tissue? Because it's multiloculated and highly vascularized with the transcriptome signature of brown adipose tissue. So UCP1 is way cranked up. And also with the presence of, stop. I messed it up, I'm sorry. With the presence of the type 2 deiodinase activity. Just give me 1 second because I messed it up royally. So this is an in vivo demonstration of the metabolic effects of human brown adipose tissue on insulin-independent carbohydrate utilization. This is very interesting, but how relevant is it in real world? So the usual so what question. And to do that, we developed this study. And in essence, first of all, as I teach at the physiology camp, get your study question first. And my study question was, is mild cold exposure sufficient to induce brown adipose tissue in humans? Does brown adipose tissue expansion results in measurable metabolic effects? Are the results reversible? And to do that, we did a fairly intense study on 5 healthy males. Now, granted, if you look at most of the studies, you have 20,000, 30,000, 50,000 study participants. But in this case, I think that it drove the message because what we wanted to do, we wanted to do a very intense observation, and we call it the Cinderella Study, whereby our study volunteers had to be back in the hospital for 4 months to stay 10 hours in their bedroom that changed the temperature dosing for 4 different periods at 24, 19, 24, back 24, and 27. And then we performed dense phenotyping with PET studies at the end of each period. And we also obtained a skeletal muscle sample and adipose tissue biopsy. So this is the scheme of the study. Each block of 30 days was a different temperature in the bedroom, and the individuals were pretty much extricated from their basement and moved. They were healthy dudes living in their mom's basement for all intents and purposes, and now they get paid to stay on at the NIH. And what we did then, we perform metabolic chamber, PET CT, blood sample, and again sample. So this is to show that while there was no correlation on the environmental temperature, we were able to modulate very precisely the temperature in the patient's bedroom. And most important, while during the day there was really no difference, so they were doing their own business, once they got into their bedroom, they were wearing two thermometers. Outside their clothing, they did achieve the difference in temperature on this panel, while under the clothing, there was really minimal difference. Just at the very end, there was a trend toward higher temperature when they were, quote, in the tropics. So what happens? This is after a 24-hour exposure at the end of the first month at room temperature. And they were young, healthy individuals with some brown adipose tissue uptake. What happens after 1 month sleeping in a colder bedroom, their brown adipose tissue more than doubled. And then back at 24 centigrades, it was a regression to baseline. And after 1 month at 27 centigrades, there was no appreciable brown adipose tissue on PET. Now, we also did lots of blood sampling during the metabolic chamber stay. And at 19 centigrades, again, we perform an oral glucose tolerance test, no difference in glucose, but a net decrease in insulin while the patients were in cold temperature, after a month in cold temperature. And also an increase in sensitivity at the adipocyte level. This is a derivative of insulin and free fatty acid. At the end of the same period, on 24 centigrades in the metabolic chambers, these results were negated. So in other words, the observations were holding true only in cold temperature. Similarly, when we look at a more refined index of insulin sensitivity, there was an increase in insulin sensitivity at 19 centigrades, and also a metabolic signature with an increase in adiponectin, which is a favorable hormone from glucose and lipid metabolism after 19 centigrades for a month, and a decrease in leptin. And with respect to the Matsuda index, there was no difference after the stay at 24 centigrades for 1 day. Again, this 19 and 24 centigrades metabolic chamber were repeated at the end of each period. So what did we learn? Human brown adipose tissue mass and function can be modulated. The activation of human brown adipose tissue has measurable metabolic effects. The effects can be clinically relevant. The modulation of brown adipose tissue is mirrored by changes in white adipose tissue transcriptome, and I had no time to show this data. The brown adipose tissue modulation results in a specific hormonal signature, again, adiponectin, leptin, and also FGF21. And the metabolic effects of brown adipose tissue expansion are measurable only during activation. And relative to this study population, the experimental model, meaning they were healthy young individuals, likely underestimates the potential benefits due to the floor effects. In other words, you cannot improve perfection, shall we say. So I'm about having been taking the helm, and the question now is what we want to do. We want to learn a bit more and exploring the interaction between adipokines, myokines, brown adipose tissue. In other words, let's go back to the bench. Just to say that the molecular physiology of brown adipose tissue or thermogenic fat is very interesting and complicated. Thermogenic fat can derive by two different lineages, either mesenchymal cells that then generate muscle, and this is the proper brown adipose tissue, the intrascapular fat, or other mesenchymal cells that then generate adipose tissue that can transdifferentiate from white and beige. Many groups have studied multiple signalings that can expand and stimulate the progression in thermogenic fat. But there is one question. What about studying the supply of substrate to the adipose tissue? In other words, why not looking at the vascularization? And Dr. Nee is going to take it from here.
Okay, it's my turn. So Dr. Celi has introduced enough information regarding how important for brown fat, especially the working mechanism for UCP1. So a lot of effort and research has been put to identify the potential regulator which can activate the brown fat activities. So far, like norepinephrine, BMP7, FGF21, irisin, and thyroid hormone have been proved to show very promising effect in both in vitro and in vivo level. But another question a lot of people have been ignored is for brown fat and beige fat, they are pretty much most vascularized tissue in your body. So this is very easy to understand because just imagine that your brown adipocyte with UCP1 expressed inside is your powerful vehicle engine. But again, you still need enough fuel supply like fatty acid and glucose to make sure your engine can run at the highest capacity and efficacy. So in terms of this process, especially for beige fat recruitment, the dramatic angiogenesis just can make sure or provide the guarantee for your engines. And also I like to share another useful information here to everyone is for basic research, especially for the metabolic disorder disease research, no investigator likes to use the female animals, although they have to, because female is not a good model for obesity. So if you feed the same cohorts of animal with a high-fat diet, you can notice that the male mice gain weight really quick, and they are very easy to develop obesity and the type 2 diabetic. But for female, it is extremely difficult. They don't know why until recently. A group find that in adipose tissue of female mice, there are higher angiogenesis factor expressed. So together with this information, it gives us a very clear indication that angiogenesis in the adipose tissue might be another target which will allow us to manipulate the brown and beige fat activity, even ends up to fighting against the metabolic disorder disease. So here we choose angiopoietin 2 as our target to verify our hypothesis. Angiopoietin 2 is a 496 amino acid long protein and belongs to angiopoietin family. And this protein has a very complicated working mechanism. When another angiopoietin family member, angiopoietin 1, is existing, angiopoietin 2 can compete binding with their common receptor tier 2 as an inhibitor. So in this case, angiopoietin 1 will work as the anti-angiogenesis factor. But in some of the particular circumstances, angiopoietin-1 itself can directly bind into the receptor to stimulate the endothelial cell proliferation. So in this case, angiopoietin-1 can work as a pro-angiogenic factor. Even angiopoietin-2 can directly bind into integrin to stimulate the endothelial cell migration as well. So in order to do our basic research, we need a research tool. Here is our research tool. We generate the NGPT2 adipose tissue specific knockout mice. So in order to generate these mice, you need two different types of transgenic mice in hand. So first is the NGPT2 flux mice. So which means you have to use molecular biology method to insert a LOXP site on NGPT2 on the mice genomes. So the LOXP is a very specific DNA sequence can be used to localize the target gene at the same time to be recognized by the Cre, which is the DNA recombinase. So and also you need another Cre carrot mice under the direction of a specific promoter. Here we use adiponectin Cre because adiponectin has been proved only expressed in adipose tissues. So when you crossbreed these two types of mice, as the offspring has both these two genes carried and expressed, especially for the Cre, only driven by the adiponectin promoter, which means the Cre only expressed in adipose tissue instead of any place else. So the Cre will recognize the LOXP site and cleave your NGPT2 like a scissor, while in all the other cells and tissues, your NGPT2 will remain untouched. So once we get the NGPT2 adipose tissue knockout mice, we start to feed them with high-fat diet for 6 months. And we are very surprised to find that compared with the sibling controls, only female knockout mice gain more weight and develop obesity, but there's no difference have been found in males. Then we decide to use the mice indirect calorimetry to characterize these mice, trying to answer why they gain more weight. So here is our PhenoMaster mice metabolic chamber. So in this chamber will allow us to characterize two individual mice at the same time and simultaneously tell the users how many oxygen consumption the mice have and how many carbon dioxide production for each individual mice, as well as food, water consumption, locomotor activity, and the body weight, as well as energy expenditure and the respiratory exchange ratio. So in which energy expenditure and RER are not directly sensor measured value, instead they are calculated value. So here's the equation behind this calorimetry. Energy expenditure equals the calorific value times the oxygen consumption rate, while the calorific value needs to calculate based on the RER. RER is the ratio value. It is the ratio between the carbon dioxide production versus the oxygen consumption. So this is a very useful value to indicate what kind of substrate your body is utilizing at the moment. For example, when carbohydrate such as glucose is utilized at the moment, you need 6 molecules of oxygen plus 1 molecule of glucose. And after oxidation, 6 molecules of carbon dioxide and 6 water plus 38 ATP will be generated. So in this case, your RER is 1. But in another case, when fatty acid is the unique substrate to be utilized in the body, your RER is 0.7. So RER is always a dynamic value of variance between 0.7 and 1. For example, when you just take a big meal or you are doing a cold exposure or doing an exercise, your RER will shift toward 1. But when you are taking a nap or you are fasting yourself, then your RER will shifting toward 0.7. Besides, our lab are also working on using this RER value to indicate the subject or the body's metabolic flexibility. I'm going to show two data, one coming from our human metabolic chamber, another one is coming from the PhenoMaster really quick. On the left, you can find when we characterize two different populations, the black curve indicate the healthy subject and the red curve indicate a metabolic disorder patient. Here is the non-alcoholic fatty liver patient. When we give them a standard meal at the same time, the healthy subject can respond to this glucose challenge very quick. So the glucose is priority utilized at the moment, so their RER increased really quick. But for the metabolic disorder patient, their response is relatively slower, and obviously they need longer time to finish to utilize the glucose from the meal. On the other side, when we compare a very healthy young mice compared with the DBDB, which is the very classical type 2 diabetic mice model in our mice metabolic chamber, we can find in the life cycle, when the mice are sleeping, not so many food eatings, the RER value has no big difference. But during the dog cycle, when the mice start to take the food, start to have the activity, the healthy young mice has a very rapid RER increase, indicate that glucose is utilized at the moment, while the DBDB mice has no changes at all compared with their life cycle. So this result tells us very well that in this DBDB mice, because of insulin resistance on glucose intolerance, the blood or the glucose in their blood could not be utilized at all, instead remaining in their circulation. So let's come back to our data. After we finish the 6 months of high-fat diet feeding, we put our mice into the metabolic chamber. So here is the over period energy expenditure measurement in both dog cycle, life cycle. So after we analyze the data, first of all, we find that compared with the controls, our knockout female mice has slightly increased the food consumption, but significant decrease of their locomotor activity. But for RER, there's no significant difference because we provide the same diet, which is the high-fat diet. So then we did the linear regression to correlate the relationship between, the energy expenditure and their body weight. We find that the regression curve slope between the genotype are very different here. So this result indicates us that our knockout mice has a significant lower energy expenditure compared with the control. This result explains very well why our mice gain weight and develop obese, because they eat more, have less activity, at the same time, they have lower energy expenditure. In terms of the result from males, we totally cannot find any difference at all, no matter in energy expenditure and also in RER or food intake, as well as activity. So it's a little bit disappointing, but it gives us a very clear direction that in the following investigations, we need to really focus on our female knockout mice instead of the males at this moment. So the first thing after we sacrifice mice is we take out their brown fat. Obviously, you can notice that there is the very significant changes from the histology perspective. So you can find here, there is the adipocyte expanding in the brown fat, which we call this phenotype as the brown fat whitening. And when we probe the brown fat biomarker with qPCR, we find both UCP1 and CDA, which are very important brown fat biomarker, are downregulated. So this result confirmed that the brown fat impairment. And also we find there is a downregulation for the AP2, or you can call it FABP4. So a report mentioned that when people develop a FABP4 knockout mice, these mice show a reduced thermogenesis and the whole body energy expenditure after cold exposure. Then we check their inguinal fat, which ideally is the best area a mice can recruit some beige fat in order to consume more glucose and fatty acid in order to keep the energy balance. So as we find that in our control groups, the female mice successfully recruit the beige fat, while in the knockout groups, they totally lost this capacity, and the adipocyte in the inguinal fat becomes really big. So we quantified the size for the adipocyte and did the frequency distribution analysis. And we find that in the control, the inguinal fat adipocyte size is majorly enriched between 400 and 800 square micrometers, while in the knockout is all about around 2,400. It's extremely big. And when we probe the thermogenic and the adipogenesis biomarker with qPCR, we find, compared with brown fat, the beige fat has even dramatic downregulation in the knockout group. And then we did the CD31, which is the biomarker of the blood vessel in the celiac cells immunohistochemistry staining. From the control group, we can find that a lot of vessels, which indicate by the red arrows here, are surrounding the beige fat in the control mice inguinal adipose tissue, which indicate by the brown arrow. While in the knockout group, because the blood vessel is barely presented, so the adipocyte in this area are experiencing unhealthy expanding. And in the paragonadal fat, we get the similar result, which we can find, again, the knockout mice has the way bigger adipocyte size compared with the controls. And when we probe the adipogenesis inflammation-related genes expression with qPCR, we find the AP2, the FABP4 is downregulated again. This result is very interesting. We're still working on to explain why or whether or not there is the direct correlation between NGPT2 and the FABP4 in adipose tissue. But a very useful information I want to share here is from a clinical study, people found in the obese population, the FABP4's expression in the adipose tissue is downregulated. Instead, the plasma FABP4 level is progressively increased according to their BMI. Then we move forward to check the mice glucose homeostasis. So first of all, from the panel A, we find that first of all, our knockout mice didn't develop hyperglycemia. And also the glucose still can be well tolerant here. But when we check the insulin level in the serum, we find the knockout mice almost have the double of the insulin levels. So when we did insulin and the glucagon double immunofluorescence staining, we find that the pancreas islet is extremely big in the knockout groups compared with the controls. So which is the very classical phenotype to indicate insulin resistance in mice model, because whenever the body tells it lost the insulin sensitivity, it will tell the pancreas to expand the pancreas islet size and also increase the number of the islets in order to release more insulins to help to control the blood glucose level. So this result tells us because of the NGPT2 deprivation in the adipose tissue, our knockout mice are developing pre-diabetic. The next thing is we characterize the liver in the knockout mice. So after I get the slides from our core facilities, I'm very surprised to find that my knockout mice are developing fatty liver, although I didn't find any difference of triglyceride level in the serum. So I'm trying to probe the fatty liver and fibrosis related gene biomarker with qPCR and find a lot of important gene which is used to control the lipid metabolism in the livers are downregulated, especially for the LXRA, the liver X receptor alpha. This is maybe the most important triggers in terms of the lipid metabolism and the cholesterol transportation in the liver. As we know, when you have LXR knockout mice and feed them with high-fat diet, these mice will develop severe steatosis in the liver. And also, when you have a chemical can inhibit the LXR activity, you will see a lot of target gene like EBP1C, fatty acid synthase, and ACC will be downregulated. And it turns out to cause the hepatic steatosis. So this is also the reason a lot of people are now using LXR activators to test their efficacy in vitro and mice models against obesity and type 2 diabetic, and has achieved very promising effect. So after I get this result, I was a little bit confused. So I asked myself two questions. First, why only female NGPT2 adipose tissue knockout mice display impaired metabolic phenotype? The second, is NGPT2 in adipose tissue more important for female or women compared with males? So as soon as I was ready to tell Dr. Celi, I want to buy some mice from Jackson Lab and kill them to characterize the NGPT2 level in the serum and in adipose tissue, a friend of Dr. Celi or a collaborator in University of Maryland delivered a very exciting information to us. So they did the clinical trial on 1,172 Amish people. And what they found is the serum NGPT2 level is strongly correlated with, inversely correlated with the BMI. And also this association is way more stronger in women compared with men. And also they find that in the serum, NGPT2 level here is significantly higher compared with men. So this result is really amazing. It just make our result on the same page, because generally speaking, if you want to keep your body shape or have better metabolic phenotype, you're better to have higher NGPT2 in your bodies. But another question we need to answer is why? Why women has higher NGPT2 level? So when I was considering the answer for these questions, the first idea coming into my brain is, whether or not the estrogen play a very important role here, because compared with men, obviously women have way higher estrogen level. So is it possible that estrogen can directly induce the NGPT2 expression in the adipose tissue, or whether or not this induction effect will disappear after menopausal happens? So in order to verify this, I designed a very simple experiment. So I did a primer culture to induce the beige adipocyte and the white adipocyte. And during this process, I used two very low dose, 10 nanomolar and 100 nanomolar estradiol to stimulate this process. And I find that the estradiol could directly induce the NGPT2 in both beige and the white adipocyte, and also has relatively dose dependent manner. Now I'm working on another project, because this data is pretty much verify my hypothesis from the in vitro level. And I'm working on to use the over-optimized mice to verify this hypothesis in vivo level. What I want to do is I'm going to do over-optimize and to deprive the endogenous estrogen level, and I want to observe how the NGPT2 level in both circulation and the adipose tissue changes. And after that, I'm trying to do an estrogen replacement therapy to see whether I can recruit the NGPT2 level in the adipose tissue after the endogenous estrogen deprivation. So here is the summary for my study. So first of all, the female NGPT2 adipose tissue non-compromised gain more weight upon the high-fat diet feeding and have lower energy expenditure as well as activities. And the second, the NGPT2 female mice show impaired brown fat activity and fail to recruit the beige fat against the high-fat diet feedings. And also, adipose tissue NGPT2 deprivation leads the mice to develop the insulin resistance and also cause the fatty liver. Finally, the NGPT2 in the circulating level are higher in women and potentially clinically more important for women compared with men in terms of the metabolism. So I'll give the slides back to Dr. Celi.
Thank you, Vin. So I'm back. So back to the bedside, and again, I always like to ask the so what question. Well, let's try to recap the story. So is this observation clinically relevant in humans? And to me, it's a resounding yes. So lack of estrogen-mediated thermogenic fat function could in part explain menopause-associated weight gain, which is a really understudied and all too obvious to ignore problem. Do we have a human disease model to investigate? Well, we can use a just pure observational large perspective course. We can use interventional studies, so elective oophorectomy, for instance, a BRCA carrier would be an ideal model if you want. And to this point, what type of study question you want to apply? Well, again, we can do a purely observational, so we can go on big data, registering multiple measurements, and perform some correlation study or association study. That is interesting per se, but also we can dig a bit further down, performing genotype-phenotype association studies. In other words, if we have an informative single nucleotide polymorphism, that can drive us to see whether the angiopoietin per se gene can explain some of the variants in, say, for instance, postmenopausal weight gain. What I'm pretty good at, though, is doing proof of concept small studies. And in essence, we can perform dense phenotyping in selective populations. And for instance, that's classical endocrine physiology. If you want to see the effect of one specific hormone, well, either you add it or you remove it. And again, we're collaborating with Dr. Karjani in OBGYN in designing a project on patients who go for elective oophorectomy. And then, is this drug targetable? Well, possibly. Definitely, global stimulation of angiopoietin-2 doesn't appear feasible because of off-target effects and potential risk for carcinogenesis. But the adipose tissue-specific delivery would be a very viable option. So in closing, back to the drawing and grant writing board. I hope we share today that it's very important having an open and continuous discussion between clinical observations and bench-based discovery that then can lead to animal models, human models, proof of concept studies, going back to preclinical studies in so-called targetable or druggable molecules target, and eventually leading to a drug development program. And then once the grant comes back and is not scored, you're not that happy, but that's the name of the game. So in closing, I would be remiss to thank our colleagues. First of all, our former colleagues and friends at the NIH that have been so important, and in particular, our late colleague and friend, Paul Lee, who led the charge in the Cinderella study. And then many colleagues, friends, students, Janine and Jared were PhD students with us. Luigi is a PhD postdoctoral fellow. And then Dr. Chen, who runs the metabolic chamber. Dr. Yavuz was our clinical counterpart in the thyroid studies. Our clinical coordinator extraordinaire. And on the cardiology side, and now on the physiology side, Antonio and Salvatore. And again, our major thank to the CRSU and our newly found collaborator, Dr. Karjani. And I thank you for your attention.
Thank you. Any questions? We have a couple of minutes left for Dr. Nee or Dr. Celi. Can I ask one? Can you tie all of this together for me with the increased risk from inflammation and obesity?
Yeah, that's an interesting question. So the inflammation in obesity is looked as in essence anoxia of adipocytes. So what happens in when the adipocytes grow larger, the surface to volume ratio is completely off, and there is a recurrent necrosis of adipocytes, which then recruit macrophages, and particularly M1s, then produce a chronic inflammatory state. So that's why we are so excited in looking at the angiogenesis in adipose tissue. So on one side, on the thermogenic path, but on the other side also, and that's something we haven't touched upon at all, on the modulation of adipocyte inflammation, related inflammation. So that's an interesting question.
Yeah, no, that's great.
Everybody awake?
I think everyone's awake. But that was just a great example of it. That was true translational medicine, observation to the bench, in vitro, in vivo, and back to patients. So congratulations on your work.
Thank you.
All right. Thank you, everybody.